Today I am very pleased to say that we have distinguished guests from Brazil, Professor Hildete, Prisco Pinheiro. She's Professor of Biostatistics at University Unicamp in Campinas, and she is belonging also to the group which is responsible for admission at University of Campinas, at university level, admission of students. She is active in Brazilian National Biostatistics Society. She is on board of directors and secretary of directorate for Brazilian Statistical Association. She was last year the president of organization committee of SINAP, which is Brazilian National Symposium of Probability and Statistics. Her degree, she graduated at University of North Carolina in Chapel Hill under direction of P.K. Sen and François Moisievich. Her thesis research involves the biostatistical issues in modeling mutation of HIV processes. But now she is in research, if I can say, of biostatistical data mining, massive data sets and applications in genomics. And today she will be talking about homogeneity tests among groups of genomic sequences. Hildete.
Thank you very much for the invitation. It is a pleasure to be here. I will try to make a small introduction of biology, even though I think I don't need here. Usually the audience is more statisticians, so I will go fast over it, just so you can have an idea the type of data set I'm working on. Okay? I will talk briefly about the Hamming distance and its decompositions. And then I will talk about the homogeneity test among groups of DNA sequence. When you have a certain kind of data set, for each individual you have a sequence, a DNA sequence. Then I will talk briefly about distance measures in microsatellite data, which is another kind of data. I will briefly explain the two different kind of data that we can work with. Then homogeneity test with this kind of data, microsatellite sequences, and I will briefly mention two applications. So if you have a DNA sequence, you know that you have a double helix sequence, and always the T pairs with A and C pairs with G. And if you are a diploid individual, you get like a pair of sequences. So you have A, C, T, G, or T. So the type of data that I will get from each individual would be a sequence of letters. Like this, sequences of letters. The difference with microsatellite data is that in some part of the genome that it's not coded for protein, or you have intron and exons. Of course, you know, in the part that it's not coded sometimes get a bunch of letters that doesn't code for anything, but it seems that has a lot of variation, and people are starting to wonder why it gets so much variation and why, if it's associated with some disease or not. So in microsatellite data or variable number of tandem repeat, what you have is repeated DNA composed by 2 to 6 base pairs. Like this one, I'll have 11 ACs, 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 like if you have 2 base pairs that are repeating. This one you have 6 repetitions. I like C, G, C, C, A. And this is repeated 6 times. So in microsatellite data, what I will see for each individual in a certain locus, what I would like to see is, sorry, thank you, is how many repetitions I have in a certain locus for each individual. Okay? So, first, thinking about the DNA sequence, only the letters, then I will briefly talk about the microsatellite data. If you have a vector xi, I indexing the individual. So for each individual, you have a sequence of letters. Can be A, C, T, or G. In this case, we have 4 categories. So the Hamming distance would be, when you look at a pair of sequence for individual I and individual J, we would like to see what is the difference between these 2 individuals. So looking at the pair of sequences, the Hamming distance would be the proportion of sites in that sequence where xi and xj differs. So the mean the measure we are talking about here is the distance measure within a group. So within a group, we will make all the pairwise comparisons. If we have a group with ng individuals for group G, we will make all the comparisons between the individuals. So this would be the distance measures between groups, within group, I'm sorry. For between groups, we will compare an individual from group G and an individual from group G prime. Okay? And then we will make all the pairwise comparisons between the individual from one group and the individual from the other group. And you'll see what's the proportion of difference you can get and divide for all the comparisons you can make, which is Ng times Ng prime. Okay? So if you get the pooled sample,
You're going to have to stand forward of that. Right in this area. Sorry.
I have to stay here?
It's just a feedback. Kind of stand in this general area. When you come back, this.
Oh, okay.
Sorry.
So if you get the pooled sample and make all the comparisons between individuals, disregarding the groups, you have this, what I call, again, D bar and 0. This is if we make the comparison disregarding the group between all the individuals we have. So, and we have n individuals in the total for the pooled sample. Okay? So, but this we can decompose as a measure within and a measure between. So, for the homogeneity test, what I'm going to do is to test whether the population, the age is for the Hamming distance for the population, and the D's are for the sample. Okay, so what I'm trying to test is if the distance that I have between groups can be expressed as the distance within and between, like the average of this. So for the null hypothesis would be that if there is no difference between groups, that this measure between groups would be just the average of the within groups distance. Okay? And the alternative hypothesis would be that this is greater than this because of some mathematical, we see that it can only be greater than or equal. So that's why the alternative hypothesis is one-sided. So as we can, doing some math, we can write down the between measure as this. Okay, twice the distance within between groups, and then minus each within group. So doing with some results of use statistics, we can get the asymptotic distribution of this test statistics, and we can actually do some application with it. We'll see later on that in my application we can even, we don't have to bother about this. Let me see if I can work with both controls here. We don't need to bother with the distribution, which is a linear combination of chi-squares, but we don't need to bother. We will just do resampling techniques like bootstrap or jackknife to do the test. Here is the other situation. If we have, instead of having the sequence of letters, you have for each individual the letters, but you're only going to count the number of repetitions you have in each site for each individual. Okay? So y here, YIGT, it's counting the number of units of repetitions of the ith copy in the jth group in a specific locus at time t. Okay? So we here, where is it? I tried the other one. So here I'm just looking at the difference between two copies, copy I and I prime, from the same group, and here if I look at different groups. Okay? Using the same idea of the Hamming distance we are using, so we can have now this distance. The difference here is that looking at microsatellite data for diploid individuals, we're always going to have 2 copies for each individual. So that's why I have a sample of T and G groups, because if I have NG individuals in group G, I would have 2 copies for each individual, so I would have 2 NG copies. Okay? And here I'm just looking at the difference squared as a measure. No, that's fine. Thank you. So the distance measures for microsatellite data, now they are similar to the Hamming distance one, but here the difference is that in the Hamming distance one we are looking at categories and see if they are different or not. Here, since we are measuring the number of repetitions, now we have a quantitative response and we can just look at the difference and square it. Okay, but the same thing goes on here with this for the pooled sample, and we can decompose the pooled sample distance in the within and between distance. Okay. The same thing happens here with we decompose the within and between measure, and we are trying to find out the asymptotic distribution of these statistics. The goal is to get a statistic with an asymptotic distribution that we can work with to actually do the test. So the homogeneity test among microsatellite sequence, we can work with this parameter here, which is actually the deviance in the expected number of mutations due to the population diverse. Because here we have to work also, if I don't know if you have worked with coalescence theory or phylogenetic tree to see how the evolution goes in a species or a gene. So behind all that theory, I also take that into account. That's why I had that T. The T, it counts for the time, because as the time goes, the genes can evolve, or the species, whatever we are working with. So the test would be if the deviance in the expected number of mutations due to population divergence in a specific locus, say L, if this is equal to the deviance in number of mutations within population, and I'm under the null hypothesis, I'm saying that this is 0. Okay, so the test statistic would be this, very similar to the one in the Hamming distance, just the way that we have the measure are a little different. I'm going to skip those formulas because this is just asymptotic theory and we don't want to worry with this right now. So let's go to the application, which is the more interesting part. For the first application, let's think about DNA sequences. So I would have case 1 with 87 sequences, which we have 58 Homo sapiens and 29 other primates. The goal here is to see if our test works. We would like to know if doing our test, if I can actually find difference between these two groups. Okay, so the interest is to find if the null hypothesis would be that there is no difference between these groups. And if we reject that hypothesis, so it means that it seems that our test is working, is actually detecting the difference. So to see that, we also got another case where we have 58, these 58 sequences, we just regrouped into 2 subgroups of 29 sequences each. And in this case, we did 2,000 bootstraps, resample 2,000 times. In case 3 is the same as case 2, we just add the bootstrap is with 10,000.
So Hildete, in the case 2, at each bootstrap repetition, there are different subsets. So when you divide 58 to 2 times 29, are they different?
No, these 58 sequences, before I started the bootstrap, I divided. But under the null hypothesis, then I have to, the distribution, the empirical distribution would be that under the null hypothesis. So under the null hypothesis, I don't know which group they are. They are on the pooled sample. Okay, but then I would do the test knowing that they are from one set is from one group and the other. I separate it first, then I will do the test. So these are the observed values of the test statistics for each case. The numbers doesn't mean that much, but if you now look at the p-values, you see that for case 1, when you do 2,000 bootstraps, you reject the hypothesis of homogeneity of the groups. That's what we wanted to detect, that the groups were actually different. It was the Homo sapiens and the other primates. Also, if we do 10,000 bootstraps, it's the same thing. We also reject. And for cases 2 and 3, we are not supposed to reject the hypothesis, so the p-value is big. It means that we don't reject the hypothesis of homogeneity of the groups, because they're actually the same group. I just divided to see if the test was working. So this is just the difference for case 1, the empirical distribution for 2,000 and 10,000. There's not much difference, so it's just to see that you don't need that much bootstrap in order to your test works. So the second application is we got data from the genetic analysis workshop. I don't know if you have heard about it. It's a workshop in the United States that I think every 2 years they have. So for each workshop they send you a set of data, and you can work with this data and then go to the workshop. The way you want to, each person, each group will analyze the data according to the goal that you think would be nice to work with the data. And later on, this data is public and you can use for any purpose you want. So I got the data from this GAW workshop number 14, and I saw that the data set would work for the methodology we were working on. So this data is from a study about alcohol, and they had all the microsatellite data according to ethnicity and alcoholism. So they had the whites, black, and others. And in the others they had Hispanics, they had the others were small groups, so that's why we grouped them here. Then they also are separated by if they were affected, if they were considered alcoholic. These are the unaffected, they're non-alcoholic with no symptoms, and these are the pure unaffected because they never drank. This group here, they had drank but they were not affected, they had no symptoms. So we had a total of 219 microsatellite loci for this data set. And the interest is to verify whether the groups defined by ethnicity and level of alcoholism are homogeneous at each locus. This test here does for each locus, it's doing a test. So you are testing at the specific locus if there is variability or not among the groups. You can do for the whole. There is a way of doing for the whole data, I mean just adding up and taking the average over all loci, but since each locus in microsatellite data has a lot of variability, they say one can just, you don't see the difference in each locus specifically if you add up and take the average. So that's why our test is taken for each locus. So because of that, we had to correct the significance level here because we are doing several tests. For each locus we do one, so it's like doing multiple comparisons, like in analysis of variance when you do multiple comparisons. So that's why we corrected by the significance level here by Bonferroni. So here, notice that when we want to look at the difference in groups, we have three groups of ethnicity here and 3 groups of alcoholism. So we don't have 2 groups anymore like we had in the first application. So we noticed that jackknife works better when you have more than 2 groups. We have done some study and we found out that when we have more than 2 groups, the best thing to do, the resampling technique, better to use is jackknife. That's why we use jackknife here. So here are it's a 95% confidence interval for our test statistic, looking at one specific locus. It's this locus over here. So we can see that according to the confidence interval, you see the 0 is way off. So the 0 is not in the interval. So we detected some difference. I'm showing here just the ones that are more interesting because we did a lot of tests with many different loci. So this is, you can find that there is a difference between the group of the ones that are affected and the non-affected, the unaffected with no symptoms. And this is the never drunk with alcohol, with the affected. And this is the never drunk and the unaffected with no symptoms. So at this locus, we could see that it seems that there is a difference between those groups. Because after you see if there is a difference between among the 3 groups, then you have to make the multiple comparison test between one and the other. That's where we corrected the significance level. For this one, this is another locus, and we could see that we noticed big difference here, but this one is below 0. Well, but there is a difference here as well, but not as much as here or here maybe. But it's something that someone that knows more about the data has to discuss about it and see what's happening with this locus. Also, this one is for difference in ethnicity. You see that there is difference, a big difference between blacks and others, also blacks and whites and others. Remember that I'm showing, it's not that there is difference everywhere. I'm showing just the loci that I found some difference. Here, the variability is much greater. You can see that the confidence intervals here are wider. So in that specific locus, the variation is high, but you can still see some difference between blacks and whites, but not here. Blacks and others, you cannot see difference anymore, neither here. Only between blacks and whites here. Here you can see difference between blacks and others, but not really between blacks and whites. And you can see between whites and others. Again, this is another locus. Blacks and white, there is no difference. And we could see that we had many different set of figures like that, but I'm showing just the ones that we could detect more difference. And according to the locus, you can see that some locus are much more variable than the others. So that's why we were doing separate tests for each locus. So here are some of the reference. This is about coalescent theory. This is about use statistic theory. It's a book. This is the paper that uses that data set, that application with the Homo sapiens and primates, and that's when we developed that that test for DNA sequences. These are all for asymptotic results of the test statistics using new statistics. And this is the last one, the one with microsatellite data. This is probably appearing, it was accepted in 2010, so it's already online, but it didn't appear in paper yet. You can get online already. And I think that's it.
Thank you.
Thank you very much.
I'm open discussion for questions. Any questions or comments? Hildete, let me ask one question. You say that you correct from Bonferroni.
Mm-hmm.
That means that you have these multiple loci tested at the same time.
Yes.
And how the p-values combined look like, and can you say something about how it discriminates between? So are these p-values or corrections already incorporated in confidence intervals that you showed?
Yes, because the overall test has like 5% level of significance. If you do the Bonferroni test, you have to divide by the number of tests you are making. If you are making, like, if we have three groups, and you're going to see if group 1 and 2 are different, 1 and 3, 2 and 3, you're making 6 comparisons. So you have to divide the level of significance 5% divided by 6. So to be significant, it's harder.
Harder because you have to...
Yeah, it's much harder to get significance when you divide the level of significance by 5%, because instead of having a p-value of 0.05, you need a p-value of 0.05 divided by 6 to be significant. Okay? I don't know if you follow. Just to try to
This is
explain.
Comparisons between the groups. Do you have any combination between loci? For example, if you
Why is that? Can I turn it off? One of them? This?
Good.
This?
Good.
Okay.
So it is loci dependent. Can you combine a few locis?
Yes, you can. Yeah, there are tests that in the paper I make a discussion about if you want to combine the test, because that was one of the questions of the referee. He asked if we want to combine all the loci, what we do, and so on. So there is a discussion about it in the paper.
Any other question or comment? If not, let's thank speaker again.